Hi everyone. Today we're going to start topic 4, which is about inheritance, how alleles are passed from biological parents into their gametes and then given to the biological children. And so question 4a says the two alleles of each gene separate. Don't forget that we carry chromosomes in pairs, homologous pairs, and each chromosome carries its own copy of the gene, which is an allele. And that's a lot of vocabulary. So let's draw this out. Since we're talking about how alleles are passed on to offspring and how those alleles are separated, of course we're talking about meiosis. So let's give ourselves a single cell, and then a single cell, and then meiosis 1, cell division. So here's your two daughter cells, and then meiosis 2, the second division event. Then you have your 4 daughter cells or your gametes. Fine. So let's pick 2 different colors for the chromosomes of my biological parents. Now, which one is my biological mother and which one is my biological father really doesn't matter. Now, what we have to remember is that the chromosomes are made of DNA. And so the section of DNA is the gene. So there's a gene that sits right there. There's a gene that sits right there. In fact, it's the same characteristic, but not necessarily the same allele. So where that gene is located is called the locus. So this is the locus of these genes, the position. So let's put a big R on this chromosome. Let's put a lowercase or a recessive R on the other one. So if you were to ask me my genotype would be big R, little r. What does that literally mean? It means that from one of my biological parents, I received a large R or a dominant R allele, while from the other parent, I received the recessive R or the lowercase r. And this is my diploid cell that starts in my gonad. So every form of cell division, mitosis and meiosis, you get semi-conservative DNA replication. And so that goes from 1 copy of the DNA to 2. So instead of this chromosome having only 2 chromatids, now it has 4. And that's why when we see chromosomes in a karyotype, they always have 4 chromatids because they are the product of semi-conservative replication. So now these are identical copies of each other. So this is an uppercase R or a dominant R, whereas over here we have the recessives or the 2 little r's, like such. And then these chromosomes line up side by side in metaphase 1. Again, still a diploid cell. You then get the attachment of the spindle fibers in anaphase 1. The spindle fibers pull, and this entire chromosome, all 4 chromatids, are pulled to the left-hand side of the cell. And of course, we are tracking those 2 dominant R's. Whereas the other side of the cell, the chromosome from my other biological parent, is pulled to the opposite side. Don't forget, this is meiosis that is occurring inside of your gonad, your ovary, or your testicle, depending on if you're a male or a female. And you are taking the alleles that you received from your biological parents and you are separating them. Segregation. This is the only time that segregation is acceptable. You are now in metaphase 2 and anaphase 2. Two spindle fibers attached to each chromosome. The spindle fibers pull during anaphase, and one copy of this chromosome is pulled to the left, one copy is pulled to the right. Same thing happens here. One copy is pulled to the left, one copy is pulled to the right. And you start seeing that you end up with the same number of chromatids here that you started with in your original cell. Except the difference is that the homologous chromosomes have been separated now. So the purple is the dominant R, or the big R, while the orange from our other biological parent has the recessive or lowercase r. Don't forget that these cells down here, these cells, these are your gametes. These are your sex cells. These are your germ cells. These are the product of meiosis. And so even though you started with a homologous pair, big R, little r, you're only going to give your biological child one of these two alleles. The whole point of meiosis is the separation or the segregation of homologous chromosomes, which then results in the separation of the genes that are on those chromosomes. So let's show fertilization where these gametes then come together. So you have the biological mother's contribution, the biological father's contribution, and the zygote. So here's the ovum, here's the sperm. So if we started with a diploid cell here with two copies of each chromosome, a homologous pair, and we ended up with only one chromosome here, that means that these cells then are haploid. These are haploid gametes. So taking that information then over here, again, the colors here, in all honesty, don't matter. Here is the biological mother's contribution with her haploid ovum, or only a half set of chromosomes. And then here's the biological father's contribution, again with his haploid gamete, his haploid sperm. And so let's say that the mother contributes a dominant R, the biological father contributes the recessive or lowercase r. So these are your biological parents here. And then here's the offspring that they produce. Here's your homologous pair of chromosomes. And the offspring receives dominant R and a recessive R. Haploid fusing or bonding by fertilization. This is a union or a fusion, however you want to say that. This is the process of fertilization. And what you end up with now, since you have a homologous pair, this cell has a full set of chromosomes, therefore it's diploid. And this is a zygote. This is why when we talk about your genotype for this individual would be big R, little r. However, when they pass on these alleles to their offspring, these alleles are segregated or separated, and they either give their child the big R or the little r. And these are the gametes. So there you go. Here's question 4A.